Thank you very much, Paul. Thanks a lot for inviting me here today. I just, before I share the screen and I stop seeing all of you, I just want to confirm that I'm very happy for you to interrupt me anytime. Just don't write me personal messages in the chat because I won't be seeing them. So in case I don't see your raised hand or anything, just text Paul, he will interrupt me, but please feel free to do that. So with that, I will start sharing my screen. And as soon as Paul confirms me that everything is okay, and that you can see my screen.
I'm still waiting. Yeah. Okay, yes,
okay.
I can confirm that I can see your screen.
Perfect. Okay, so as Paul said, we are going to have this lecture is about protocells. And so I figured that the first question you might have is, why do we need a Protocells 101 lecture at all? Why should we care about protocells when we think about how life started on our planet, or whether life is possible or present elsewhere in the universe? And so I figured I would use a very common, let's say, approach in talking about this, which is presenting you the two common approaches that are normally used in the research for the origin of life. So when we try to understand how life started on our planet, the first approach that is normally followed by some researchers, especially in the biology area and field, is the so-called top-down approach, which basically means that when we want to understand how life started on our planet, we look at modern biology and we sort of try to go backwards from there, trying to understand what is required, what is needed for life to exist. The second approach would be the so-called bottom-up approach, which starts from simple molecules, simple ingredients that are non-living per se, but when combined together, they make more complex systems that potentially could help us understanding what are those needed and required components that can make life. So in this context, combining these two different approaches, in the middle, the conjunction between these two approaches is where protocells lie. And the reason is that if we think about the bottom-up approach as the approach of prebiotic chemistry that starts from very simple chemicals and try to combine them into something more complex that could lead towards life, and we think about modern biology and through the understanding of modern biology, we think about simplifying that. We can think about, we find that the combination of these two, or the conjunction between these two, is where minimal life forms could lie. And so protocells could help us understanding how, what kind of design principles are at the basis of the emergence of life. But not only that, because once we have these design principles that can tell us a bit about what kind of, how life could have started on this planet, once we have these design principles, we could also have the basis to study different kinds of living systems, which means, for example, understanding whether different chemicals could yield the same kind of living systems or different types of living systems. And this could inform us not only, of course, of what life, of how life started on our planet, but also what we could be looking for, or what we could find on different planets. So I figured that after this explanation of why we need this lecture, the next point to address was what we mean by the word protocell. And I think protocell is a word that is definitely very used, sometimes also abused in a lot of different fields. You might have read about protocells in the context of synthetic biology, in the context of nanomedicine, nanotechnology, and definitely in the context of origin of life. So I figured that what we needed was a definition for that, and luckily enough, someone did that, so that I don't have to be the one finding a definition for this. And so I looked at the Encyclopedia of Astrobiology, and I looked at the definition that a prebiotic systems chemist who works in Spain, Kepa Ruiz Mirazzo, provided for the word protocell. So according to him, protocells are any experimental or theoretical models that involves a self-assembled compartment linked to chemical processes. And these experimental or theoretical models are aimed at explaining how modern cells may have emerged. We'll go through all of these bits that I've just mentioned in the next slides, but what I want to make clear is that protocells are basically models of modern living systems, of modern cells. So if we look at what a modern cell is, that modern cells are quite complex systems. They are systems that host a multitude of different chemical reactions. And these chemical reactions take place in different compartments that are localized inside a cell. So when we think about how a primitive cell or protocell could look like, what we think about is something along those lines, much more simplified. So when we think about a primitive cell, we think about something that resembles a cell that has maybe less chemical reactions occurring within them, or maybe less molecules hosted inside them. But more or less, the idea is that a primitive cell, as I was saying before, reading the definition, is a model of a modern cell. So what do we need?
Can I just ask a quick question?
Yes.
Yeah. So if a protocell is always a model, does that mean that you're not really talking about what would have been there on the early Earth? You're just talking about models of what would have been there. Whatever would have been there would be different than what properly would be spoken of as a protocell.
I guess that it's difficult to know. Of course, we know it's difficult to know what exactly was there, right? And what exactly was available there. So in this sense, I think model here means that it's a simplified version, a very simplified version of that. And because it's not just a theoretical model, but it could also be an experimental model, we can definitely try to build that, let's say, using the chemicals that we think might have been available, or simplifying something that is nowadays. So I guess that the word model here doesn't mean something too abstract. It's not only a theoretical, it doesn't have only a theoretical meaning. I'm not sure if that answers your question.
Another question. Does a protocell have to contain genetic material, or is it just a compartment for chemical reactions?
Yeah, so if you allow me, I will address this question in the probably next few slides.
Excellent.
But if I don't, just remind me of this question later.
I will ask it again. All right.
So as this question that I was just asked leads towards, the right question at this point is to understand what we need to make a protocell. So if we go back to this very simple schematic of what a protocell or a primitive cell might be, what we need to make a primitive cell or a protocell is something that contains something else. So we have some material, a primitive content that we want included into a primitive compartment. And this means that a primitive cell can very much simplified as just a combination of these two components, of these two ingredients, a compartment that hosts some content. And you can see that now I'm being very, very vague, and that's intentional because there's a lot that can affect primitive cells and their formation and their composition. And what's important is that among all the different chemicals and all the different molecules that were likely or available on early Earth, the structure, the composition, the shape of the molecule will determine if that molecule will be part of the primitive content or of the primitive compartment. But we'll get to that in a few slides, because what I think is really needed right now is to understand why protocells are important in the context of origin of life. Why can't we just study chemical reactions occurring in solution? Why can't we just study chemical reactions occurring in an Erlenmeyer flask, for example, or in a round bottom flask? Well, what are the evolutionary advantages that protocells bring compared to reactions that occur in solution? Well, first of all, when we think about protocells, we are thinking about a system that is physically separated from the solution. So we have, as we said before, a primitive content that is hosted inside a primitive compartment. And that primitive compartment is a physical boundary that separates the inside from the outside. This means that that compartment helps defining the identity of the system that we have. Second, we can imagine that if we have molecules in solution versus if we have molecules inside one of these protocells, one of these compartments, what we can imagine is that the presence of a protocell forces molecules to be in the same physical space compared to what would happen in solution. Protocells ensures that reactants, that molecules are kept close enough. And by ensuring that, they can help, for example, the reactivity between different molecules. The third point is that protocells can prevent population extinction. So if we think, for example, that we have different molecules in solution, and it's possible that some molecule would be more reactive, so would tend to undergo chemical reactions more efficiently than other molecules. If we think, for example, about these in a solution context, so no protocells around, then the molecules that tend to react more efficiently will always tend to react more efficiently. And if the molecules that react less efficiently are more useful than the others, you would just lose them because they will never react because of the presence of those that react more. While if you have protocells, you can imagine to have, for example, a protocell that hosts those useful but less reactive molecules, the protocell that hosts those more reactive but less useful molecules. And then even if the molecules that are more reactive will still tend to react more efficiently, the others will still have the opportunity to react because now they are no longer competing with the first ones because they are physically separated. So if we go back to what was seen before, and so how primitive cells are made and what we need to make primitive cells, I anticipated that I said before that the molecules that were available on early Earth will be, of course, very varied. So there will be molecules that have different structures, different composition, different shape, and these properties will ensure the molecule to be either part of the primitive content or the primitive compartment. But what I think is very important here is, and it's the first take-home message for today, is that these molecules could have well arise from the same scenario, in the same kind of environment from the same kind of chemistry. So the next slides I'm going to show you are probably slides that most of you are already familiar with, because they describe a scenario that is called the cyanosulfidic scenario, which is a scenario that was proposed a few years ago by John Sutherland, who works at the LMB in Cambridge. And the whole idea behind this cyanosulfidic scenario is that all the building blocks of life, all the ingredients that we need to make up our cells might have a common origin. And they all derive from a very simple molecule, which is drawn here, which is called hydrogen cyanide. Now, I won't go into the chemistry behind this scenario, also because previous 101 lecture already covered hopefully all of what I'm going to show you. But the idea is that from hydrogen cyanide through different reactions, one could get the building blocks of nucleic acids, the genetic polymers that are present in our cells. And these are depicted here in orange and are called nucleotides. From hydrogen cyanide, one could derive amino acids. These are the building blocks of proteins, again, one of the main components of our cells. And from hydrogen cyanide, one could make lipids. And these are the main components, for example, of membranes, cellular membranes, which are that boundary that I was mentioning before defines modern cells identity. So put everything together, don't look at the chemistry behind here, but this is just to show that from all these building blocks, all the building blocks of life, amino acids, nucleotides, and lipids, can have a common origin and could have originated from one very simple molecule, hydrogen cyanide, on early Earth. Now, I'm showing you that all of these molecules could have originated from hydrogen cyanide, and this means that all of these molecules could have potentially been available on early Earth. And so, because these are the building blocks of modern cells, they likely contributed to forming very simplified versions of cells, and so primitive cells. But as I was saying before, all of these molecules, the participation of all of these molecules either into the primitive content or the primitive compartment that make up a primitive cell would depend on the structure, the composition, or the shape of these molecules. And we'll go into that in a moment. So when we think about primitive cells, as I said, we have two ingredients, the container, the compartment, and the content. And so the first question that one wants to address when we talk about primitive cells is, what do we need to make a compartment? Second question would be, what can compartments do? And the third question would be, what can compartments host? What kind of chemistry can occur inside compartments that can make up a primitive cell? So I'll first start with the first question, and feel free to interrupt me anytime I am unclear about what I'm going to say. So what do we mean by compartments? So what kind of compartments were available on early Earth? What kind of compartments would form, and which kind of molecules we would need to form these compartments? Well, there are very different types of, a large variety of compartments that could have formed on early Earth. One of the important points to make here is that the interactions that would occur between molecules that can drive the formation of compartments are mostly intermolecular forces, which means that these are forces that occur between different molecules and that not necessarily involve the formation of a bond, a chemical bond, a covalent bond between two molecules, but these are interactions that could still be strong enough to hold together the molecules and keep them close enough in order to form stable compartments. So of compartments that have been studied in the context of origin of life. If you've read something about primitive cells and compartments in this context, you probably are more familiar with liposomes and coacervates, and we will talk in a moment about all the different names that can be used to define exactly the same structure. You might be less familiar.
Oh, sorry to interrupt.
Yes.
Alex Archibald has a question.
Yeah. Sorry to interrupt, Claudia. This is really fascinating. Okay. It's kind of right pushing my chemistry. So as an atmospheric scientist, could I think of as an aerosol as a compartment? Because I think of an aerosol having a side which faces the atmosphere, some sort of maybe a surfactant type, molecule, which and then an aqueous core or something like that. And is an aerosol a type of compartment in that context?
Yeah. So definitely aerosols. I haven't mentioned them, but definitely aerosols are another type of systems that is studied in the context of favoring, enabling, catalyzing chemical reactions. And so in the same sense, it could be used or it could be studied in this context as well in terms of protocells, let's say.
Cool, thanks.
You're welcome. Okay, so so I was saying that you might probably be more familiar with liposomes or coacervates, and I'll give you a bit more about them in a moment. But you might have read, for example, about hydrogels and liquid crystals. So basically, all the molecules that were available on early Earth could interact with each other, and depending on their structure, their size, if they are polymers or not, if they are capable of reacting or not, if they are charged, for example, so if they have positive or negative charges or not, they could interact with each other and form structures that could be called supramolecular structures, which means that we are at a different level of complexity, and we are thinking about how molecules, a large number of molecules, can behave and assemble into something bigger. So as I mentioned, but if you have questions, just let me know about these other systems. As I mentioned, I'm going to focus on liposomes and coacervates, mostly because they are the most studied systems in origin of life research in the context of protocells. And the first thing I want to say, I will start with liposomes. And the first thing I want to say is that you are probably all very, very confused, because every time even if you've read just one paper on protocells, you probably have found a lot of different names that refer to exactly the same structure and that are all different. So I've written here liposomes, but to describe exactly the same system, you could have read vesicles or vesicles, for example, exactly the same thing. You might have read for example, micelles, this would be a slightly different kind of structure. I'll show you in a moment. You might have read membranes. So unfortunately, these words, and I'm guilty of this as well, unfortunately, all of these words are interchangeably used in publications, public talks, and everything. And I'm so sorry about that. But the important thing is that liposomes and vesicles refer to exactly the same kind of structure. Membrane, when we talk about membranes, we are mostly referring just to the container, to the these. I don't know if you're seeing my cursor, but basically only this layer that separates the inside from the outside, without focus on the inner part. So these are the most common words that you would find to describe this kind of system. If you've heard of any other word, just let me know. We can try to clarify that as well. So how do we form liposomes? Well, what happens in modern cells is that modern cells have, as I mentioned before, their boundary is what's called a cellular membrane. And cellular membranes are made of a large variety of molecules. The most common ones are lipids. So again, you might have heard different words to refer exactly to the same kind of chemical. You might have heard amphiphile. You might have heard the terms hydrophobic, hydrophilic. I'll try to explain this in a moment. So this is what would be called a lipid molecule. A lipid molecule is a molecule that is made of two parts. You can see in the upper part of this drawing that there are quite a lot of oxygen atoms. There's a charge here, for example. This makes this part of the molecule particularly capable of interacting with water. And for this reason, this part of the molecule is described as hydrophilic because it loves water. And this would represent the head group of a lipid molecule. This part that is represented here, that is colored here in gray, would be, as you can see, it doesn't have oxygen atoms, and it's just an aliphatic chain. And this means that this part of the molecule would not be able to interact with water, for example, would not be able to form hydrogen bonds with water. And so it doesn't like water that much. And for this reason, this part of the molecule would be called hydrophobic, which means it doesn't like water. And so because of this, and this would represent the tail of our lipid molecule. And so because these molecules have two components, a hydrophilic and a hydrophobic parts, that's why they are also called amphiphiles, because they have two sides, basically two parts within one, just one molecule. So you might have heard amphiphile to describe this kind of system, refers exactly to the same thing. So this is the kind of lipid that you might find in modern membranes. And what happens when you have these lipids is because what I've just said, that is that the gray part doesn't like water. The molecule would assemble so that it avoids for the gray part to interact with water and to be exposed to water. And this means that these molecules would form a layer that is actually a bilayer, and that here there are two layers of the same molecule, one flipped 180 degrees. And this is a lipid bilayer that, to minimize the exposure to water on the extreme parts, would assemble into a sphere. That is what would be a membrane. So what kind? Yes.
So first off, what makes that molecule hydrophobic and hydrophilic practically? Like,
Yeah,
what
so
is it about it that does?
Yeah, so it's what I was mentioning before about the presence of oxygens or the absence of oxygen sample. The presence of oxygen means that the molecule can, in this case, this part of the molecule, the upper part of the molecule, can interact with water forming hydrogen bonds. And so because it can interact with water forming hydrogen bonds, this part of the molecule would like to be exposed to water. So if the molecule was just it, then you would have a molecule that is soluble in water and that's it. But because it has this component that is reported in gray, which is just aliphatic chains, this part doesn't have ways to interact with water in a favorable way, let's say, because it cannot form hydrogen bonds. And so it would not like to interact with water, and that's why it's called hydrophobic. It can remind you of the structures of fat. So if you think about olive oil, this is exactly the kind of system, this is similar to the kind of molecules that would form olive oil. And so you know that olive oil definitely doesn't like water. So if you mix them, they would stay separated.
Cool.
Does that answer the question?
Yeah, I think so. A couple of other questions. Do all aliphatic molecules do this? And also, are there things that can live in between, in the sort of space in between the sort of tails of these aliphatic molecules?
Yeah, so this is a representation of lipids that are found in our membranes. And this can show that, and by looking at this, you can see that you have a relatively long chains. So you have relatively long aliphatic chains, the gray part, and then you have this sort of bulky head group that is reported in red. But if you change the shape or the size of these molecules, for example, if you shorten a lot the aliphatic part, then the molecule might lose its character of amphiphilicity, because the hydrophobic part would be too short, for example, and so it would no longer want to avoid water, and so it would no longer self-assemble into a lipid bilayer. Okay. I forgot what was the second.
Oh, and can you get things in between the?
Yeah, so definitely this is of interest for a lot of different kind of chemistries. And so if you have molecules that are completely hydrophobic, they would like to be you would find them localized in the hydrophobic part that is in between the head groups. So in the internal part of this bilayer, you would find you could localize, you could put molecules that are very hydrophobic. You can also have, if you form this membrane and then you add molecules that again have an amphiphilic character, they would insert themselves and expose the hydrophobic part to the internal layer and the hydrophilic part to the external part, and so on. So definitely you can use these as reactors, for example, for other kind of reactions that don't like water, for example.
Thank you.
Answer? Okay. So when we think about, so this is what happens in modern cells. What about early Earth? What about the kind of lipids that could have been present there? Would they do exactly the same? Well, first, the kind of lipids that would have been available on early Earth would be different if we look at them from a chemical perspective. So here you have an example of a lipid that could have been available on early Earth. And the reason I'm saying that is because aliphatic chains have been found in interstellar molecular clouds. And also these molecules, like the one I've shown here, have been found in meteorites. So assuming that these molecules could have been present on early Earth, what we can see is that this molecule, even if it's much smaller compared to the one that you have on the right, the chain, there's only one aliphatic chain, it's much shorter compared to the one on the right, and the head group is also very small compared to the one that you have on the right. Still, you can imagine that if I explained well enough what we mean by hydrophobic and hydrophilic, you would recognize that, again, this molecule has a hydrophobic component, which is depicted in gray, and a hydrophilic component, which is this pale pink that is on the top. And so this kind of lipid could behave in the same way as modern lipids. Now, how do they behave effectively? Well, if we have this molecule and we just put that in water, as we said before, it might not like water. And what happens is that if we put this molecule in water, it would phase separate exactly in the same way as oil and water does, because this molecule, when it's in this form and this chemical structure, cannot interact with water sufficiently in order to form lipid membranes, and so it would just form oil droplets floating around in water. But when instead, if we look at this head group here, when we have a more alkaline pH, which means that the solution is alkaline, and so this molecule would no longer have what is called a carboxylic acid head group, which is this specific chemical moiety here that has this carbon with a double bond with an oxygen and then a single bond with an OH group. When we take this molecule and we put that in an alkaline solution, what happens to this molecule is that this molecule would lose the hydrogen and would get a negative charge on one of the two oxygens. This molecule now, that is what we would call deprotonated, because it doesn't have anymore that proton on the oxygen, can now form a different kind of structure that is called a micelle. That is exactly what happens when you wash your hands with soap. You might have read about how soap works now that we've spent an entire year washing our hands every 10 minutes. So probably what I'm going to say is redundant, but basically what happens is that this molecule has a sort of conical shape because it has this relatively long tail and a bigger head group. And so it has a conical shape, and the best way this molecule can organize itself in order to avoid the exposure of the hydrophobic part to water is by forming these spherical systems with all the tails pointing inwards and the head groups pointing outwards. And this is, just to go back to what Paul was saying before, what happens, this is something that could be used if you want, for example, perform chemical reactions with fully hydrophobic chemicals, because they would take place inside the micelle.
Can I ask a question about
Yes.
that? A couple questions, actually. One is, are there any modern organisms that make their membranes from such simple lipids or use this kind of chemistry?
You mean these kind of lipids, very short?
Yes.
Not that I know.
Okay. And the other sort of question is, wouldn't it be really difficult to make such long hydrocarbon tails based on that hydrogen cyanide chemistry mentioned earlier?
Yeah, so what we have seen with the cyanosulfidic scenario, exploiting the cyanosulfidic scenario, is that you can make these kind of systems long up to C10, so 10 carbons in their aliphatic chain. And this is more or less the kind of size that you are seeing here. but there are other kinds of chemistries. For example, there are some chemical pathways, one of which is the Fischer-Tropsch synthesis. These are chemical pathways that can be used to make longer chain aliphatic compounds. And it has been proposed that these kind of chemistries could have occurred on early Earth as well. And so it is not impossible to think that it is not that these kind of molecules could have formed, although it would be very nice if we could implement the synthesis of more complex lipids with longer chains within the cyanosulfidic scenario, I think.
All right, thank you.
Okay. So if you look at the pictures we have here, we still haven't made membranes. So I said that when we have the carboxylic acid in its protonated form, so when it's a carboxylic acid on the head group, which happens at acidic pH, we have oil droplets. When we have a higher pH, we have a deprotonated form of the carboxylic acid, and we have micelles. So what happens in between? Well, actually, in between happens exactly what we hope that, what we need, which is that we can form lipid membranes. And the reason we can form lipid membranes is that basically when we have a pH that is intermediate between the two pHs that we've just discussed about, what happens is that you would have one molecule per sort. So you would have one molecule that has the hydrogen attached to the oxygen and one molecule that doesn't. And basically what these two molecules would do would be sharing that hydrogen. And so there would be this bridging hydrogen between two molecules. And so now when we think about the shape of this molecule, now we have two molecules that are sharing this hydrogen, and the shape of this molecule is now much more cylindrical rather than conical. And the shape of this molecule favors the formation of lipid bilayers. And so if we are in the right conditions, these lipid bilayers would form this sort of spherical system that we called liposomes. How would that be relevant for origin of life research? Well, if we can think about ways to produce these chemicals, these lipids on early Earth, then we can imagine, we know that it's very easy to make liposomes. If you imagine to have, for example, these molecules that are called fatty acids because of the fat part that is the hydrophobic chain, and acid because of the presence of a carboxylic acid on their head group. If we imagine to have these molecules on early Earth on a surface, it's just sufficient to put some water, to add some water to this surface for these molecules to self-assemble into liposomes. And even if you imagine evaporation and rehydration cycles or wet-dry cycles or these kind of oscillations, these solutions of these liposomes would just get dry, and the lipid would form a film again on the surface, and then it's just sufficient to rehydrate. So add again some water to reform these liposomes. So if no one has questions on liposomes,
one last quick question. What is the pH that you're trying to aim for typically for?
So it's very flexible in the sense that if you take one specific lipid, then it would have a one specific, a relatively narrow range of pH values that you want to make these liposomes. But if we think about that in a prebiotic chemistry context and origin of life context, we definitely wouldn't have purified lipids on early Earth. We would have a mixture of them. And it was shown that mixtures of lipids form liposomes that are now mixed. So there are a lot of lipids, different lipids on the liposomes. And this mixture actually enhance the stability at different pH values. So mixed liposomes can survive in a wider range of pH. Most of the time, however, we are thinking about a range of pH that is around the neutral pH, not too strict with that, but that goes, let's say, between 6 and 10, more or less.
Okay.
And just to give you a comparison with modern lipids can form membranes at pH up to around pH 5, then it starts to be pH 4 already starts to be complicated. So we are not that far away from that pH range in which modern lipids can form membranes.
Okay, thanks.
Okay. So I mentioned that there was another kind of compartment that I wanted to talk about, and this would be coacervates. So if you remember the cyanosulfidic scenario picture scheme that I showed you before, I told you that we could envision a common origin for the 3 building blocks of life: lipids, nucleotides, and amino acids. And you might know that nucleotides and amino acids are the building blocks of nucleic acids and proteins, and as such, they can polymerize, so they can form long chains of amino acids, which would then become peptides and then proteins, or of nucleic acids from nucleotides. So the reason I'm telling you this is because coacervates form from polymers, mostly from polymers, so long chains of molecules, for example, amino acids or long chains of nucleotides that that can interact through electrostatic interactions. This means that these polymers that we are looking at would be negatively or positively charged. And so one could imagine that the negative or the positive charge could come, for example, from the side chain of peptide of amino acids, or from nucleotides, where we know that the phosphate group that holds nucleotides together in nucleic acids have negative charges. So if we imagine to have polymers of amino acids or nucleotides on early Earth, they would have positive or negative charges we would have positive or negative charges on these polymers, and the presence of these positive and negative charges make these polymers attracted to each other, and by attracting each other, they would interact, and so they would form some kind of very dense structures that are very concentrated in these polymers compared to what happens in solution. So here, in a different way compared to what we said about liposomes, you don't have a chemical boundary that separates the inside from the outside, but here you have very dense structures that phase separate. So this is called liquid-liquid phase separation. So you have basically very dense liquid structures that are in solution and that are very concentrated in your polymers compared to what happens to what's present in solution. So with this, I'll just conclude this first part of the talk, which was a long one, so don't feel too worried about how long this talk is going to take. But as you might have guessed, this was mostly work that was performed in the origin of life context by people who are interested in understanding what is needed to make a compartment, with a focus on the chemistry behind that. So what kind of molecules were available and that could self-assemble into compartments. And these are just some people, I'm sure I've left someone out, but these are just some people that are contributing to answer these questions. The second part that I mentioned before is, okay, we have a compartment. What can compartments do? And I will not be too detailed on this, but if you have questions, just feel free to ask. Well, what can compartments do once they are in solution? Well, we've said before, and it was sort of the whole point of this talk, that compartments can host molecules, so they can concentrate molecule inside of them. But compartments can also grow. This is a cycle, a primitive cell cycle that was proposed in 2001 by Shostak, Bartel, and Luisi, that envisioned that compartments, in this case, these are liposomes, can grow by being fed by other compartments. And at some point, these compartments can divide when they become too big and so thermodynamically unstable. Something else that compartments can do, for example, is localize molecules on their surface, because we've seen before, for example, that some of these lipids could have negative charges. And so if they have negative charges, maybe they can interact with something that has a positive charge. And then if you have something that has a positive charge that is localized to the membrane, then you can that can interact with something that is negatively charged. And here again, this is something that looks very complex. Sorry about that. That was developed by the Shostak lab. Basically, they took compartments and they put some positive charges on the compartment, on the membrane, so that that positive charge could interact with RNA that is negatively charged. So this is just to show you that compartments can also be used to localize molecules on their surface and not only host molecules within them. Compartments can be combined. We've talked about two different types of compartments. We've talked about liposomes, we've talked about coacervates, and actually it was recently shown that coacervates could be formed inside liposomes, and they could also be destroyed and degraded still within these liposomes in response to external stimuli, for example, pH. So if you have questions about this, just feel free to ask. But these are some of the people that are looking into what compartments can do. And clearly, you might notice that there are more people interested in understanding what compartments can do rather than in the kind of molecules that could have been available to form them in the first place. And so these are, again, some of the people that are trying to understand how, what can compartments do, what's their behavior, and how they can mimic the behavior of modern compartments.
So I do have a couple questions. How do you get across the compartment, say, to get food inside?
That's an amazing question. And so the very simple answer I can give you is that the kind of liposomes that I've shown before that are made of very short chain fatty acids, so the very short lipid that I was showing before as a primitive lipid, those membranes are relatively leaky, which means that molecules can relatively easily go through. Here I'm talking about small molecules, so for example, activated nucleotides or amino acids, in some cases metal ions. For more complex molecules, like for example, oligonucleotides or peptides, that's more complex, and it's an answer that we don't have yet.
Okay. And do these compartments interact with minerals?
So there has been quite a lot of, well, not quite a lot, but there has been research interested in understanding first whether mineral surfaces can help in the formation of these compartments in the first place. This is again something that has been studied quite a lot, and also in Jack Shostak group. So Martin Angsik published a science paper in 2003 maybe, that was about using mineral surfaces to help in the self-assembly of lipid membranes. And so in this regard, minerals have been studied with lipid membranes. But I would say that minerals sometimes are more seen as an alternative to compartmentalization, rather, or a different type of compartmentalization rather than interacting with compartments, because of course, especially porous minerals can host and can absorb molecules in a similar way that one would envision compartments do. So in this sense, they are sort of seen in a contrasting way rather than interacting with each other.
Okay, thank you.
Okay, so I'll move on if you don't have any.
Oh yes, please.
This is just the last part of the talk. What can compartments host? And here I could have shown you a lot of different chemistry, but I decided to be very light on chemistry. So we said before that a protocell is made of a compartment, a primitive compartment that hosts primitive content. What kind of primitive content has been studied so far in the context of protocells and origin of life? Well, the first example that one can think of is the polymerization of RNA. There's quite a lot of work, for example, performed by the Shostak group, but not only the Shostak group, that describes the possibility of having RNA polymerization, so the formation of nucleic acids starting from single monomers occurring inside membranes, lipid membranes. There's been a bit of research also into polymerization occurring inside coacervates, not as much as within liposomes, but this is definitely the first thing that people look at because of the relevance of RNA in terms of genetic polymer, polymer. The second example is the synthesis of peptides. There have been few examples of studies that focus on the formation of peptides within compartments, again, mostly within within liposomes. And the third one is the study of the formation and activity of metal complexes. So systems that are based on metal ions bound to different kind of molecules and the within primitive compartments to study their potential as catalysts in a sort of by looking at them as ancestors of metalloproteins that are quite common in our cells. But definitely among these three types of chemical reactions and chemical processes that have been studied inside liposomes or coacervates inside of compartments, polymerization of RNA is definitely the most well studied. So I just want to conclude with one bit. We said, I've shown you before this cyanosulfidic nitrogen, and I said that there are these shows the possible common origin for all the building blocks of life. But by what I've said, you might have realized that there isn't really a strong distinction between all of these building blocks of life. It's not that lipids only form compartments and nucleotides and amino acids only take part to the primitive content of the protocells. I mentioned, for example, that polymers of amino acids and nucleotides can form coacervates, so they can form compartments themselves. But something else that has just started to be explored, and I think has a lot of potential, is the combination of all of these molecules that have the potential to interact with each other. And so there's been a recent study that we did in Jan Netherlands group, where we looked at how all of these components interact with one another. And so we could see that not only the compartments should be seen as containers, inert containers. They are not just flasks where we are performing our reaction. They can participate into the reactions that are taking place within them. And so we should study what's the effect of the chemistry that we are performing inside our compartments on the compartments themselves, and how compartments can enhance or take part into this chemistry. So with this, we have the people who are mostly, or who have mostly worked on this last part. So what can compartments host? As I mentioned, Shostak, but also a lot of other people who are trying to understand what kind of chemical processes can be hosted within compartments, and how the compartments are affected by the chemistry encapsulated within them. So this leads to the last question, to the last slide, which is what's next? What's next in the field of protocells in the context of origin of life, and not only? Well, these are just some of the questions I could think of when I was preparing this talk, but of course there are many more that hopefully will get addressed and answered in the next years. So one question could be, once we've made a primitive cell, what happens to all the other primitive cells around them? How do these primitive cells interact with one another? How these primitive cells interact with the environment around them? I've shown you at the beginning that modern cells are quite complex. They have multiple subcompartments inside them. So how can primitive subcompartments be generated and sustained? And then one of the most important questions at the basis of primitive cells work is, how can compartments support and help enable Darwinian evolution? So definitely there are a lot of questions that hopefully will be answered soon. I just want to thank all the people I've worked with in the past. And this is a very, very ancient picture from when it was allowed to be unmasked and be all together and enjoy some beer and crisps, but hopefully this will happen soon again. And I want to thank you, all of you, for sticking with me for an hour.
All right. Please join me in thanking Claudia for an absolutely wonderful and inspiring overview of protocells and all the forms that they might take. We certainly have some time for questions, and certainly for this, I would encourage people to just raise their hands and feel free to ask anything about anything that you've seen. And it may well be that most of the questions were. Oh,
ask one.
Craig, please go for it.
What's the closest we've got to a metabolically active protocell?
We are not close at all. No, in the sense that, so compared to the study on polymerization of nucleic acids, for example, there's much less research on metabolic cycles, for example, and most of this research is quite recent and has not been studied yet in liposomes or other kind of compartments. So I don't have yet an answer for that. Hopefully soon.
Cool. Be cool to see. So, Peter Peterson has a question.
Hi, thank you for the talk. I was just wondering how much of the chemistry that you showed is possible to recreate with heavier elements?
I don't have an answer for this. I've never thought about that. What kind of heavier elements are you thinking about?
I'm thinking, well, if you could replace carbon, hydrogen, and oxygen, and nitrogen with just like one element down the periodic table in the respective column.
So I think it's a great question, and the answer is that we don't know a lot about that. What I can say though is that definitely there are some chemical reactions which would be, let's say, element dependent, but there are some some properties of the molecules that I've shown that could be present in other kind of molecules as well. So it might be very interesting to understand whether it's mostly, for example, for the formation of compartments, is mostly the shape of the molecule rather than its composition that can drive the self-assembly of molecules, but of these of membranes. For example, I've shown lipids that have a phosphorus atom, but there are a lot of other kinds of lipids that can make the same kind of membrane structures, and they could be made of sulfur, for example. So there is some kind of variability that you can change and play a bit with the molecules and the chemistry of these structures. But there isn't a lot of study in this direction, so it would be difficult to answer at this point.
Thank you.
So Nick has a question right now, and then there's a really nice question in the chat by Clancy Jiang, who may want to ask that in person or not. I'm not exactly sure, but Nick, please ask your question.
Thanks very much for the presentation, really enjoyed it. My question was, how do liposomes, how do they behave under relatively high ionic strength conditions? So when salt concentration gets high, you mentioned the importance of wet-dry cycles, I guess, and concentrating and then rehydrating them. But then, of course, the inorganic components will then be carried along with that. So I'm just kind of curious as to what's known about salinity and things like that.
Yeah, great question. So as I was mentioning before, when I answered to Paul's question about the pH range in which they can survive, it really depends on the composition of the lipid membranes. So if we think about the lipid that I showed before, that is called decanoic acid because it has 10 carbons and the carboxylic group. That kind of membranes made of just that molecule would be relatively unstable. You would need very high concentrations of that lipid to make membranes, and they wouldn't tolerate at all divalent cations in solution, and also they wouldn't tolerate concentrations of sodium chloride higher than 150 millimolar maybe. But if you play with the composition of the membranes, so if you start including other kind of surfactants, other kind of lipids, and this is another word that I didn't mention before that is sometimes used to define this kind of, to describe this kind of molecules. So if you mix them with other kind of lipids, then the stability can increase. And it has been shown that when you have mixtures of fatty acids, or even if you include other kind of head groups like alcohols, for example, the stability to ionic strength increases, so you can go to higher concentrations. There have been studies that showed, for example, that the seawater concentrations are acceptable in for some kind of liposomes. So even higher concentrations of salts can be tolerated. The problem about divalent cations, which would be magnesium, calcium, for example, is a problem that is not solved yet, though, because the problem there is a different one, and it's that the head group of those molecules, which is a carboxylate, can interact and bind divalent cations, which means cations that have two positive charges, and so as such, they form a complex that precipitates out of solution, so that the lipid is no longer available to form the liposome, and so that would destroy the membrane. So that's a question. That's a big problem that we haven't yet solved.
Okay, great, thank you.
Thank you, Claudia. So Clancy does have a question. I'm going to preempt it with a bit of a question of my own so that I can understand the answers to Clancy's question. You showed this great artistic picture from some of Jack Shostak's work showing the sort of liposome gets big and then it divides. Is this a computational model or a theoretical model, or is this an actual thing that's been seen experimentally?
Yeah, so this is something that they've seen experimentally.
Okay.
So they've taken liposomes made of these fatty acids that I've shown, so of these primitive lipids, and they've seen that if you feed them with more fatty acids, so if you add fatty acid in solution, these vesicles tend to grow. And then what they've seen is that at some point, these very large liposomes become unstable. And by just introducing some, they can induce the division of these very big, large liposomes, for example, by agitating the solution, or by inducing chemical modification on to some extent to the lipids. So they have seen that, and they've followed that by microscopy. So they could see the formation of the division and in smaller vesicles or liposomes. So this is definitely something that has been looked at. What hasn't been done yet, and this also maybe goes back to the question that Craig asked, is that so in the injects view, the growth and division of vesicles or liposomes should be coupled with the replication of RNA, so that you can have daughter primitive cells that can keep replicating and making more complex versions. So this is something that has not been achieved, even though the two systems work separately. So RNA can be polymerized and vesicles or liposomes can grow and divide. The combination of the two has not been shown yet.
I see. So with that sort of preliminary question out of the way, which will be really helpful to me anyway, Clancy, would you like to ask your question?
Oh, yeah, fantastic talk. So my question is also related to the stability of the liposome. As you say, they grow larger, and to a certain degree, they will spontaneously divide. And by adding phosphate to it, so phosphorylizing the hydrophilic end of the liposome, would that like enhance the stability of this liposome?
So I'm smiling just because this is something that I'm studying right now. So great question. Now, what I can say is that specifically, so in general, there hasn't been yet a lot of work performed to understand exactly how this works. I mean, the growing part is relatively clear. The division part is a bit less clear. And there's definitely people that are trying to understand that by playing with the components in solution. So for example, there's a very recent paper that was published by Sherif Mensi, where they mixed together two different populations of liposomes that are made of different lipids, and they see that the addition of one to the other, to the first to the second, drives the growth, and the second to the first drives the division. So there's a lot behind that still needs to be addressed and understood. Specifically about phosphate, sorry, I just wanted to say that specifically about phosphate. Phosphate is a very peculiar ion, and it's a sort of legend in the origin of life research and protocell research is that membrane liposomes made in the presence of phosphates are very unstable. No one knows why. No one knows exactly what's happening when you have phosphate ions in solution. So this is why I was smiling, because this is something that I'm trying to address right now. But if you're interested about that, we can definitely talk about that.
Yeah, you did mention the replication of RNA does not coincide with the division of the liposome. That if you are replicating RNA, you got to form the phosphate sugar backbone. So you have to have phosphate in the solution.
Yeah.
And so that wouldn't that say if the membrane is unstable in the presence of phosphate, could that be the triggering point of the liposome division?
It might be. I don't have an answer for that. It might be. So definitely the present. So definitely RNA polymerization works inside liposomes, and it doesn't make them breaking apart, for example. But as far as I know, they haven't tried to combine that with the growth and division process. So they haven't tried to feed the protocells to make them growing and so on. So I don't know whether the problems would be at the next step when they want to try to grow and divide them or not. Definitely the presence of phosphates have an effect on the stability of membranes, so that's for sure. Whether it's less strong, the effect when the phosphate is in the nucleic acid moiety rather than just free in solution, this is something that I hope I'll address soon.
Exciting.
All right. Are there any more questions? From Clark Baker.
God, oh, I'm invisible. Here I am. This is a very naive question, but do we see any sort of protocells forming naturally in nature, or is that something we don't see? And if not, what's sort of stopping them? Is it the conditions aren't necessarily correct now, or is it some sort of like biological process blocking, sort of the timescale is longer than some sort of biological process that's happening?
So when you say if this happens in nature, do you mean if we observe formation of liposomes or membranes in our cells, or whether it would happen if we have just lipids in our sea salt?
Yeah, so if you I don't know, sort of you just left a pond of water sitting somewhere and you sort of scooped it up and you looked, could you sort of find like a membrane with chemistry happening inside of it?
So I guess that if the body of water that you are exploring had a layer of lipid molecules available there, you would definitely observe the formation of lipid membranes. That's for sure.
Right. Okay.
So it really depends whether you would have those molecules in those lipids in the first place, and I guess that also the kind of chemistry that we are studying in prebiotic chemistry, when we think about what kind of chemical pathways could have led to the formation of nucleotides, amino acids, and lipids, it's difficult to know whether that it's difficult to know the relevance of those chemical reactions in nowadays conditions would be completely different to what it was the relevance on early Earth.
The difference is sort of like the complexity of the lipids, which we would see now would be.
Also, for example, the cyanosulfidic scenario that I mentioned before is heavily requires UV light, and the UV light that it requires would be a UV light that does not permeate and does not reach the surface right now, because on early Earth there wasn't the ozone layer, for example, right? So that kind of chemistry would not occur in the conditions that we have not today on our planet, and so that's why I was saying those molecules might not be available right now, but if they were, they would form compartments. Yeah.
Right. Okay. No, brilliant. Thank you.
Thanks. All right. If there are no further questions, let's thank Claudia again. Thank you very, very much for a wonderful talk and overview. The last thing that I'm going to say, if you are interested in further talks about a whole bunch of different things, there's a Cambridge Exoplanet and Life Day that is coming up on the 8th of June, between 10 a.m. and 2 p.m. I'm going to put a link for that and other events coming up from the initiative here. And please, if you are interested, just drop me a line at my email. And I can give you the link to be added to the relevant mailing lists. We will be having a bunch of other events coming up, and really look forward to seeing a lot of you there. So thank you all for attending and hope to see you again soon.
Thanks a lot, Paul, again.
All
Thanks,
right.
Claudia. That was great.
I'm happy you liked that.
It was good.
Wonderful. All right. See you all.
Bye.
Bye.